Hello, my name is Maxwell Johnson, and today I will be presenting on genome editing. So, what is genome editing and how can we use it? So, genome editing is the precise process of altering the DNA of an organism. So, this involves adding, removing, or altering DNA. Anytime you add or remove DNA, generally speaking, that's genome editing. Genome editing shows a lot of potential, mainly in agriculture and medicine. I've found that a lot of the benefits with genome editing are mainly in those two fields. So, for example, you could increase the yield of a crop, or you could do really solid cancer research with genome editing. And we have three main methods we're going to discuss today, and those are ZFNs, TALENs, and CRISPR-Cas9. So, starting it off with ZFNs, zinc finger nucleases are engineered in order to be used for targeted genome modification. They have binding domains that need to basically specifically designed, and they also have a non-specific DNA cleavage domain that we get from the flockal endonuclease. So this cleavage domain has to dimerize in order for DNA cleavage to be achieved. It absolutely has to. That's the only way cleavage is going to happen. With talons, it's very similar to ZFNs. Basically, transcription activator-like effector nucleases are engineered for genome modification. Each nuclease still contains the custom-made DNA binding domain and the DNA cleavage flockal endonuclease. Cleavage still only occurs when the DNA cleave dimerizes, right? So very similar to ZFNs. The main difference we have, however, is with the spacer region. So in talons, the spacer region is somewhere between 6 to 40 base pairs, whereas with ZFNs, this is around 5 to 7 base pairs, so drastically different spacer regions. CRISPR-Cas9 is definitely the most well-known method of genome editing. It was adapted from a defense mechanism found in bacteria. So first, DNA from an infection is incorporated to a CRISPR locus, and then the CRISPR loci is transcribed into large RNA, like very, very large RNA fragments, and then those get processed into short cRNA fragments or CRISPR-derived RNAs. And then these cRNAs can be, they can interact with Cas9 nucleases and kind of recruit them to be complementary to viral DNA sequences. I've always thought of this, I don't know if this is correct, but I've always thought of it as like, oh, you're like taking it over. I found that helps me a lot. So what exactly can we do with CRISPR-Cas9? We can use it for a larger number of very interesting things. For example, at Colossal Biosciences, scientists extracted DNA from direwolf fossil thousands of years old. The DNA was sequenced in order to be able to build out a full genome for the direwolf, and then using CRISPR, scientists identified 14 genes that were key, and they combined those with a modern gray wolf genome to be able to make a mixed embryo. And this new embryo was implanted into domestic dogs who just kind of served as surrogates, and the result was the birth of Romulus, Remus, and Khaleesi, I believe is how you say it. But yeah, 3 direwolf pups that, I mean, we haven't seen direwolves in thousands of years. So yeah. And then I got most of this from the Gen 3020 Lecture 19 slides. I think I actually got all of it from the Gen 3020 Lecture 19 slides. Yeah. And that is that.